Hi, welcome for today's lesson. Before we start the lesson, we will take a quick review of mitosis using a video presentation. Regeneration of body parts is just one of the things that depend on mitosis. So mitosis actually is used to replace worn out cells, to produce new cells, and also for growth. It's also used for growth. So let's go have a quick review. So mitosis actually is the division of a cell into two cells, or the division of, so as you divide the cell in mitosis, so in mitosis we only divide the cell into two, for example, to replace or for regeneration of body parts, or for growth, or producing new cells. So from one new cell, we will produce two identical cells. So the first thing to be identical will be the DNA. So the DNA will be duplicated and will produce new cells. So after duplication of DNA, we have now two daughter nuclei. Mitosis actually has four stages or four phases. The first one is prophase, the second one is metaphase, the third one is anaphase, and the fourth one is telophase. So these are the four stages that mitosis goes through. Then we have the last one, cytokinesis, that is the division of the cytoplasm. So what you see here is actually what we call cleavage furrow, or cleavage furrow, which normally occurs in animals. In plants, we said that we normally will have a cell plate developing within the cell that is dividing before a cell wall develops on it. Before division, the cell prepares to divide by growing or increasing in size, and also by duplicating the DNA material. So that is interphase. During interphase, that is what the cell does, preparation. We saw that interphase has G1 and S stages and G2 stages. Then the cell enters the prophase. In prophase, the chromatin actually condense to form chromosomes. They will condense to form chromosomes. So what we have here are chromosomes. So these chromosomes have what we call sister chromatids. This being one of the sister chromatid, this is another one. And sister chromatid are exact copies of each other. Then the nucleolus, the cell wall will rupture at this moment or disintegrate. Then the centrioles centrosomes will move in, and spindle fibers will form. So centrioles will go to the poles, and then spindle fibers will come through. Then we'll enter metaphase. In metaphase, the microtubules or the spindle fibers will actually align the chromosomes in the center of the cell. That is why in the equatorial plate or the center of the cell. Then sister chromatids will actually which are being held by the centromere, will split. They will be split loose and they will be pulled by the kinetochore, and they will split loose. When they split loose, they have entered anaphase. So during anaphase, they will be pulled towards the poles. During anaphase, they are pulled towards the pole. As you can see from the video here, they are pulled towards the pole. So in the equator here, there is nothing. After that, we will enter another phase, which is now telophase. We expect, you can make prediction of what will happen in telophase. It may be, you can pause the video and think about it, think what will happen. Then telophase comes in. During telophase, the spindle fibers disintegrate. The chromosomes are again loosened to form chromatins, and the nuclear envelope is reformed. So you can see the nuclear envelope has been reformed. Then we enter cytokinesis, where a cleavage furrow will split the cytoplasm into two in animals. In plants, we said we have, so here we are already having two Dora cells. We have two nucleus. In plants, it is different. They will develop a plate here. That is what we said. Then, of course, it will split and form two Dora cells, just like in plants. And that is what we called mitosis. So today we will be looking at something else actually. We will be looking at how mitosis is controlled. So today we will be looking at regulation of the cell cycle. The key concepts here include cell cycle regulation is necessary for healthy growth. So think about, maybe sometimes you go to watch a movie where people are dealing with elements in nature. They might bring back like dinosaurs or new fangled robots, and how you have noticed that at the end of the movie actually becomes always very scary. The reason is that things go out of control. So these robots take over, or the dinosaurs start eating humans. So if cells growth goes out of control in your body, the result can be even scarier. So cancer is what we call uncontrolled cell growth and results from many factors. Cancer is uncontrolled cell growth. Cancer is uncontrolled cell growth and results from many factors. Results from many factors. So we'll be looking at some of these factors as we discuss through that affect the cell cycle. That affect the cell cycle. So if factors are affecting the cell cycle, then they might lead into cancerous cell. So how does your body regulate all the millions of cells, cell divisions happening in your body? Think about it. Think about how your body will actually be able to regulate all the millions of cell divisions happening within your body. That is what we are going to look at today. So we have internal and external factors that regulate cell division. We have internal and external factors. And external factors regulate cell division. So cell division is regulated by internal and external factors. Cell division is regulated by internal and external factors, sorry. So this is a very I wanna just highlight it. That's all. So internal and external factors regulate cell division. So both external and internal factors regulate the cell cycle in eukaryotic cells. In eukaryotic cells, both external and internal factors regulate cell division, regulate cell cycle in eukaryotic cells. In eukaryotic cells. So external factors, just as the name suggests, come from the outside of the cell. They include messages from nearby cells and from distant part of the body. So external factors include messages from nearby cells, messages from nearby cells, and from distant parts of the organism's body, and from distant parts of the organism's body. Organism's body. Internal factors come from inside the cell. These are external. Internal factors come from inside of the cell. Internal factors come from the inside of the cell. They come from the inside of the cell and include several types of molecules found in the cytoplasm, several types of factors several types of molecules, that is, found in the cytoplasm. So we will be looking at them in details. So both types of factors work together to help your body control the process of cell division. So we will first of all look at the external factors. So external factors that normally control cell cycle, external factors that control cell cycle are so we are having here external factors, So external that help regulate cycle include physical and chemical signals. They include physical and chemical signals. Include physical and chemical signals. So you might be asking yourself, what are these signals that are physical in this case, and which ones are chemical? So one example of physical signal is cell-cell contact. So if we are writing here physical, include cell-cell contact. Cell-cell contact. Which means when a cell grows and touches onto other cells, then it ceases to continue growing. So most animal cells grown in the laboratory form a single layer on the bottom of the culture dish. They will form a single layer on the bottom of a culture dish, like you can see here. They just form a single layer. They form a single layer. Once a cell touches other cells, it stops
Cancer
dividing.
is a group of over 100 diseases characterized by abnormal, uncontrolled cell growth.
It stops dividing. Once a cell touches other cells, it stops dividing. So we can say, once a cell touches other cells, it stops dividing. So it will not continue dividing after it has touched the other cell. So the exact reason for this phenomenon is unknown. One hypothesis suggests that receptors on the neighboring cell bind to each other and cause the cell's cytoskeletons to form structures that may block the signals that trigger growth. So we will stop here, and in the next video we will progress with the same discussion.